last week, I know this is a long time ago because it was Wednesday of last week when we did this lesson, okay? We talked about the modes of nutrition. Anyone remember what they were?
Phototroph.
Close. Photo-autotroph had all the right parts. Photo-autotroph and
Chemio-autotroph, something like that.
Chemo-heterotroph. Yeah. Okay. So those are the two modes of nutrition. The photo-autotrophs use photosynthesis. Okay, and the chemoheterotrophs have to consume organic material, okay, in order to get their sugars. Basically, once they have their sugars, either by photosynthesis or eating stuff, okay, then from there they can use the same process, cellular respiration, in order to gain their energy. Okay. Glucose is obviously the fuel, okay, and how they get their fuel is their mode of nutrition. Those are the two types. Then we talked about how those different modes of nutrition can lead to differences in the structure of the cell. And that was kind of this slide right here, okay, where we were looking at a plant cell and an animal cell side by side. All right? And we said that because plants are photoautotrophs, they are typically going to have chloroplasts. Okay, that organelle that contains the chlorophyll where all the photosynthesis takes place. Okay, they are going to have a cell wall to help support the shape of the plant and a water vacuole to assist in that same thing. Keep the shape of the plant from the inside, pushing against the cell wall. Okay, because plants don't move. All right, this isn't the Lord of the Rings. Okay, they're not ants. They're not going to pick up and go. Okay, break stuff. Well, would it be cool? It only depends if they're your friend. If they're not your friend, it isn't as cool. I mean, ask Sauron. It probably wasn't his. They probably weren't his friend. He probably didn't think that was good. Okay? He had it coming, but you know. So if we're looking at the chemoheterotroph, that would be the animal cell. Okay? In order to be a chemoheterotroph, you have to obviously have organelles capable of digesting food. So lysosomes, peroxisomes. Okay? Things that contain digestive enzymes that can break down food particles. And they also have the centrioles that help them with cell division because they don't have a wall. And so the shape of the animal cell can change radically, and if that happens during cell division, some of the DNA might end up in the wrong place and the cells will die. All right, so they have to have those structures as well. So those were the differences in the structures of the cells due to the different modes of nutrition. Then we labeled our cell diagrams, and that's where we left off. Today, we're going to pick up with the next thing, which I will apologize right now, is going to be a ton of me talking and you listening. This is what I don't like about biology. There's a lot of me talking and you listening. It's like empty vessel teaching, okay? And I don't like that very much. The diagrams. Anybody else not get a cell diagram? All right, Alex, you need one too?
Yes.
Okay, if you haven't already done so, make sure you watch.
Sorry, Mr. Mayor, can I also have one? I lost mine.
Why don't I just grab the whole pile then?
Oh, never mind, I have it. Sorry.
Five more? Maybe. I feel like Devin's gonna end up in the Brady Club. Okay, so what we're gonna go over today, guys, is we are gonna go over what compartmentalization is, okay, which is a really long word but a really simple definition. Okay, we're going to look at the role of each organelle. All right, so this is the lesson where you learn what each part of the cell does. That will be important because on Thursday's quiz, not tomorrow's quiz, Thursday's quiz, you will get a diagram of a cell, and you will have to label and give the functions of the parts I decide to ask. Okay? So we'll be going over that. Okay, we'll look at how all of those roles go together to help with the functioning of the cell. Okay, we'll look at how the cell carries out all those functions, and we're going to look at cell transport. Okay, and there are the big three methods of cell transport that we will look at: diffusion, osmosis, and active transport. Okay, so it's a lot today. Okay, unfortunately, on a Monday, just the way it worked out. Okay. All right, questions before we get started? Take a deep breath and then never stop. The rest of the class. Okay, compartmentalization, important term. Okay, so compartmentalization, okay, is present only in what we call eukaryotic cells. Now, compartmentalization means very simply that the cell is broken up into compartments. Okay? Each one of those compartments is an organelle. So the nucleus is a compartment. The lysosome is a compartment. Okay? The mitochondria is another compartment. And compartmentalization is a major advantage for any cell/organism whose cells have it. Because compartmentalization allows processes that may not be compatible with each other to occur at the same time under ideal conditions. Okay, so compartmentalization simply means that there are different compartments or environments within the cell. That would be each of the organelles in there. Okay, and inside each one of those organelles are the perfect conditions for the job that organelle does to be carried out. Okay? Only eukaryotic cells, that is cells that have organelles in the nucleus, okay, obviously have compartmentalization. There are actually two kinds of cells, eukaryotic, that's what we have, and prokaryotic. Prokaryotic cells are generally bacteria. Okay? They are much smaller, simpler, they don't have any organelles. All of the processes that they carry out, which are all the same processes that your cell carries out, have to take place in the same room. Okay? Everyone kind of follow me on that? Okay. Is that going to be good or bad? They all got to be carried out in the same room. Yeah, it's generally bad. Okay? I mean, think about all the things a cell does. It's got a separate environment to hold all of its precious, fragile DNA. Then it's got another environment where food gets digested, and another environment where sugar gets burned in order to release its energy. Okay? Do you want to have all three of those processes happen in the same place? Because it's possible you might digest your DNA or burn it. Okay? So, I mean, literally not that, but okay, you kind of get the idea. The conditions are not going to be ideal for anything. It would kind of be, if you wanted an analogy, kind of like the one I have here. In your house, you generally have three separate temperature environments. The environment that most of your house is at, 21 degrees or so. Okay? You run your furnace, you keep your house at 21 degrees or so. Okay? Your refrigerator, which is usually somewhere between 4 and 6 degrees Celsius, where the cooler temperatures inhibit the growth of mold and bacteria and keep your food from spoiling for a short period of time. And then you have your freezer, okay, which is kept usually around minus 15 degrees Celsius, which can prevent food spoilage long term. Okay, we're talking months. Okay, not years. You don't want to eat something that's been in there for years. Even though it hasn't spoiled, it'll be freezing. Okay, now those are the three separate environments. That's a eukaryotic cell. Okay, those three jobs are not compatible with each other. Okay, a prokaryotic cell has to do those three jobs in the same place. So it would be like keeping your house at about 10 degrees Celsius, because you'd have to find a happy medium for those three jobs. Are you comfortable at 10 degrees Celsius? Not without a coat on, probably. Okay? Is your food going to spoil fairly fast? And you have no ability to store it long term at 10 degrees. Okay? But that's what a prokaryotic cell that doesn't have compartmentalization has to deal with. Okay? It has to deal with jobs not compatible with each other being done in the same place. Okay? Not great. That's why you never see a large prokaryote. It's why you never see a multicellular prokaryote. Okay? Without the complexity of compartmentalization, it is impossible to carry out all those jobs at the same time. Okay? At least well. They all get done, but not well. Okay? Everyone all right with that? It's kind of like being a jack of all trades and a master of none. All right, so eukaryotic cells have compartmentalization. Prokaryotic cells, and incidentally, if you're wondering how that's spelled, okay, prokaryotic, okay, those are cells that don't have compartmentalization. Okay, everyone understand what compartmentalization is? Okay, compartmentalization is going to be, one of those concepts that probably ends up in a multiple choice question somewhere. Okay. All right. Anybody need that slide anymore? Okay, so compartmentalization allowed cells to become larger. Okay, prokaryotes have no compartments and are much smaller than eukaryotes. Okay, and more specialized in their functioning. Eukaryotic cells are far more specialized or have the ability to specialize, where prokaryotes do not. So a prokaryotic cell would be like this bacterium here. Right? You can see in there this snarled looking stuff, that's the DNA. It's just out there. Okay? There's no membrane around it to protect it. It's just out in the cytoplasm with everything else. Okay? Nothing there to keep it safe. Right? Which is why bacteria can mutate so quickly. Okay? Their bacteria is often, their DNA is very often altered or easily altered, okay? Which can cause pretty major changes in their ability. And when I was in university, that was one of the things, E. coli was one of the things we used in our genetics classes all the time. Okay? Because if you put E. coli bacteria under a UV light for like 30 seconds, a small percentage of them will become immune to penicillin, like that. Okay? Scary, but true. Okay? The whole thing with radiation causing mutations is totally true, making you have claws and being able to do weird things like in the X-Men. No, okay, but for a bacterium, yeah, they totally become like an X bacteria, and they suddenly become immune to antibiotics. Okay, so it doesn't take much, all right, and they are fairly simple, and they're also fairly small. If we're looking at size comparisons here, a bacterium is going to be maybe 2 micrometers. Maybe a large one might be 4 or 5. That's it. Very, very small. Okay. A typical eukaryotic cell doesn't even fit in the picture. Okay. A typical eukaryotic cell is going to be, even a small one is probably going to be somewhere in the neighborhood of 30 micrometers. Okay. We looked at some under the microscope, like the amoeba and the paramecium, that are large. Okay. Those are about as large as single-celled eukaryotic organisms get, several hundred micrometers. You have very few cells in your body that are that big. Most of yours are between 30 and 80, and much smaller, okay? But much bigger than a bacteria, or a virus, which is less than a micrometer in size. Okay? Everybody follow me so far? Okay, so that's compartmentalization. Okay, let's talk about the compartments and the parts of the cell. Okay, so DNA. Its job is to it's the instructions for making materials, and it is stored in the nucleus. So what is the job of the nucleus? Okay, well, the job of the nucleus is to store DNA. Okay? And then we're going to cross this out right here, because it implies that protein synthesis is controlled from the nucleus when it is controlled. I guess red's the best color for that. From the nucleolus. Why did I put a U in there? Hang on. I made a spelling mistake here. I think that was supposed to be an L, and then I made 2 L's. Nucleus. There. Nucleolus. Nucleolus. There we go. Okay, so protein synthesis is controlled from the nucleolus. DNA is stored in the nucleus. And honestly, you don't need to know much more about it than that. Okay? The single job of the nucleus is to store the DNA. And in the environment of the nucleus, the DNA is everywhere. Okay? When the cell is dividing and you can actually see like individual chromosomes, that is the only time DNA actually looks like that. Okay? Once the cell is finished dividing and all the DNA goes back into the nucleus, it completely unravels. Okay, so inside the nucleus, it would be like someone took a ball of yarn, completely unraveled it, and threw it in a pile. Okay, that's what your DNA would look like inside of the nucleus, completely unraveled. All right, so imagine the task of being able to know where the code for adrenaline is versus where the code for estrogen is, okay, in that DNA. Being able to get those two things, okay, that's the job of the nucleolus. Find it, copy it, control protein synthesis. Okay, so nucleus stores DNA, okay, and nucleolus, okay, produces copies of the DNA and controls protein synthesis. Okay, now, you don't need to understand how protein synthesis works at a very complex level for Science 10. Okay, so I'm going to explain it, and then I'll tell you that you probably don't need to know it in even that level of detail. Okay, so what happens is your cells need to make stuff. Okay, hormones are a good example because they're proteins, and your cells have to make them all the time. Okay, so if your cell needs to make a certain protein, okay, your nucleolus will find the part of the DNA that codes for that, and it will send a special enzyme, okay, called RNA transcriptase. Okay, and that enzyme pulls the DNA strands apart. Right, because DNA is double-stranded. Now, because the strands, and I think you learned this in Science 9, are complementary, right? A always goes with T, G always goes with C. You only actually need to copy one side, because you know what the other side would be, because they're always complementary. So the RNA transcriptase pulls the DNA apart and copies one side of the strand and then puts it back together. Right? That copied piece, okay, so that would be this piece right here, okay, is the copy, goes out through the nuclear pores, okay, those little holes that we talked about in the nuclear membrane. That copy of the DNA, that RNA, okay, because it's not double-stranded, it's just RNA, goes to a ribosome, and the ribosome reads it. And each part of it codes for a different amino acid. Those amino acids get put together, and that forms a protein. Okay? Now, I told you a few things in there that you don't need to know. Here's the story of protein synthesis for Science 10. Ready? Nucleolus copies DNA. Copied DNA goes to the ribosome. Ribosome makes protein. Okay? That's the extent to which you need to understand protein synthesis for Science 10. Okay? Because it tells you the role of the nucleolus and the ribosome. Okay. Now, how often is this happening? All the time. Okay? There could be lots of different proteins being made concurrently within your cell. So DNA is constantly being pulled apart, put back together, pulled apart, put back together. Okay? Luckily, it's fairly durable. Okay? All right, so we've covered the nucleus stores DNA, nucleolus copies DNA and controls protein synthesis, and ribosomes make protein. Okay. Why don't they just make the proteins in the nucleus?
Because the environment?
Well, because of the environment, but also because what can't get into the nucleus? Anything. Yeah. Okay. So it's really hard to make proteins if the stuff you need to make proteins can't get in there. Right? So access to raw materials, as it says here in the last sentence, okay, is better in the cytoplasm. That's why we copy the DNA and send the copy out. Okay? All the raw materials are in the cytoplasm, so that's where we do it. Okay.
Can I have some water?
Yeah. Anybody need this slide anymore? All right. Now, endoplasmic reticulum. There are two kinds, rough and smooth. Okay? They look considerably different. Right? Smooth ER, under an electron microscope, kind of just looks like this. Rough ER looks much more organized. Okay? Kind of folded over. Okay, so this kind of diagram here, okay, you can see that it's folds and folds and folds with ribosomes stuck to it. Okay, so all of these little black dots in this diagram, each one of those is a ribosome. Okay, and that is greatly enlarged through the use of an electron microscope. Okay? We are talking that those ribosomal subunits are fractions of a micrometer each. Okay? They are exceptionally tiny. Right? The smooth ER, by comparison, doesn't look like really anything because it's just kind of everywhere. Right? It's very much under a light microscope, almost impossible to see. Because it's just transparent. Okay, so talking about what the ER does. All right, the rough ER just transports protein. Okay, it's got ribosomes all over it. The ribosomes are always making protein. The rough ER's job is to transport that protein. Okay, the smooth ER, on the other hand, has lots of jobs. Okay? And that's why I said no cell diagram ever does justice to how much smooth ER there is inside of a cell. Okay? It fills up a lot of the cell, you just don't see it. Okay? So the smooth ER, first job is that it makes lipids. Lipids is another word for fats. Okay? This is an extremely important job because all membranes, the nuclear membrane, the cell membrane, the membranes of the mitochondria and all the other organelles are all made of fats. So the smooth ER makes fats to help repair membranes because membranes are always getting damaged. Okay? So smooth ER makes lipids. Okay, so lipids are synthesized, okay, in repairing membranes. Complex carbohydrates can be broken down in the smooth ER. Okay? Complex carbohydrates are things like starch. Okay? So if you had a big plate of poutine, okay, all the starch in the fries, okay, is going to get broken down in there, okay, and then sent to the mitochondria, okay, as simpler sugars. Your lysosome breaks down like proteins and fats. Okay? The other thing that gets broken down here is toxins. Okay, so we've got complex carbohydrates being broken down. Okay, drugs and poisons are detoxified. Okay, so drugs and poisons get detoxified within the smooth ER. This is important because the smooth ER has its own membrane, which it can rapidly repair because it makes the stuff that repairs membranes. Okay, and that isolates the toxins. If the toxins are kept in the smooth ER and they're detoxified in there, then their toxic effects are limited to just the smooth ER, which can rapidly repair itself. Okay, so they're generally detoxified there. Can all things be broken down as detoxified? No. Okay, there are some toxins that cannot be broken down and detoxified, and there are some that can be. Okay, so typically, like simple things, could be broken down if you, had some, harsh preservatives or something like that in your food. Okay, your smooth ER can break those things down. If you, not that any of you would do this because you're too young, but if you went out drinking, okay, and you had a lot of alcohol in your system, the smooth ER is the part that's going to break that down. Okay, now the problem with breaking down toxins is usually the products of that are less toxic, but they're not great, okay? Which is why if you've ingested something that is somewhat toxic, the process of breaking it down doesn't generally feel very good.
Hangovers.
A hangover, exactly. Okay? The reason you feel that way is because your body is getting rid of all the toxic crap you put in it, okay? And some of those products are in and of themselves toxic, okay? And there's only a couple of routes out of the body for toxins, okay? And your body will use them in order to get rid of them. Okay, you think about what part of your body is responsible for detoxifying poisons. It's your liver. It's right here. Okay, it's your largest organ, other than your skin. Okay, and so when it's detoxifying the poisons kind of end up in your bloodstream and can go out with your kidneys, filtered through your kidneys, or they'll end up in your intestines because there's a big duct that comes down, okay, called the common bile duct. It comes down from your liver and goes right into your upper small intestine, which is right where you don't feel good. Okay? And it's because all that stuff is going in there and it's very irritating and can make you vomit or have it come out the other end. Okay? But either way, it's trying to get rid of really nasty stuff. Okay? So those are kind of the byproducts. So if I was looking at a liver cell, a liver cell would probably have more smooth ER than maybe a nerve cell would, okay, or a muscle cell. They don't need them as much. They're not doing that job as much. Okay, that sort of makes sense? Okay, and the last thing is that hormones can be produced there. Now, these are special hormones. They are fat soluble. Hormones could be produced there. So, does the smooth ER have lots of jobs? It does. On a quiz or a test, how many of them would I expect you to write down?
2.
1. So on Thursday, when I say name and give the function, if smooth ER is labeled, one of those functions is given. Okay? All right. Now, here's the thing now. I don't want anyone to just go and memorize only one thing that the smooth ER does. Because in a multiple choice question on a test, I could say, you know, the creation of fat-soluble hormones is the job of A, B, C, or D. And if you've only memorized one function and it wasn't that one, you're kind of cooked on that. Okay? But on a quiz where I'm asking you to give the function, I'm only requiring one. All right. Questions on that? Okay, and then like we said, lipids can be quickly moved through the smooth ER to the places in the cell because the smooth ER goes everywhere. Okay, it's in contact with just about everything. All right, and so if something needs its membrane repaired, it's got to contact with the smooth ER and it can ship the lipids there and get that membrane repaired and toot sweet. I think that's the right expression. I don't know. I don't do French very well. All right. Okay, Golgi apparatus. So the Golgi apparatus is responsible for packaging material and sending it to the membrane. That's typically going to be waste products, but it can also be products produced by the cell for export. Okay, that would be hormones and things like that. Okay, if this was the adrenal gland, okay, cells of the adrenal gland, then the Golgi apparatus would be packaging a lot of adrenaline, okay, to be exported to the cell membrane. Now, if you're looking at these pictures here, okay, you'll see that almost all of them have this label on them: cis face, trans face. Okay, cis face, trans face, and even in this electron microscope picture, you see the same thing: cis face down here, trans face here up above. Now, you'll notice that the trans face always looks a bit different than the cis face. Okay, coming off of the trans face are bubbles. Okay, you can really see it here in the electron microscope picture. And like there's a bubble, there's a bubble, there's a bubble, there's a bubble. There's all kinds of bubbles coming off. This right here is the Golgi apparatus. Okay? The cis face is just a bunch of small stuff coming in. Okay? So the cis face is receiving. The trans face is shipping. Okay? So things come in one side, they come in the cis face, okay, and they go out the trans face. And when they come out, okay, they are encased in a vesicle, which is just made of membrane. Okay, so they've just made a little transport vesicle, okay, a little bag essentially that holds the material inside it, okay, and that material is made of exactly the same material as the cell membrane. So once it merges with the cell membrane, it just, whatever's inside just pops out. Okay, now it's out of the cell. Okay, and it's either into the interstitial space, that's the fluid between the cells, or it's directly into the bloodstream and being carried elsewhere. Okay, everyone kind of follow me on that one? Okay, what you really need to know is that the Golgi apparatus packages material for export from the cell. Okay, at a basic level, that's what you need to know. Okay, knowing the cis face and trans face and that kind of stuff is good, but at the basic level, you need to know that the Golgi apparatus packages material and sends it away. Okay, questions on the Golgi? All right, the lysosome we talked about already, okay? Inside the lysosome there will be strong digestive enzymes that will allow material to be digested, broken down into simpler things, and typically what we're looking at there are fats and proteins because the smoothie R takes care of the carbohydrates, okay? All right, so inside of the lysosome, this is kind of what a lysosome looks like. Inside there are hydrolytic enzymes. Those are things that can basically break stuff down quickly. Right? So a lysosome, this is actually, this diagram should say that this is a peroxisome. Okay, and then this is a lysosome. Okay? A lysosome actually has the food in it, a peroxisome does not. I'm never going to ask the difference. So if you're like scrambling to write that down, it's not a big deal. Okay, so at a basic level, what does the lysosome do? Breaks down food. Yeah, breaks down food. It can also be part of what's called autophagy. Okay, autophagy is basically the cell being the ultimate recycler. Right? If an organelle or something like that within the cell ceases to function properly, it will be digested and treated as food. Okay? So it will kind of cannibalize itself if something stops functioning properly. Okay? And we also said the other day that a lysosome can also be part of kind of a self-destruct mechanism. Okay? If the cell begins to fail, becomes malignant, and the cell can recognize that, it will cause autolysis, okay, to happen, okay? And that means that it will basically digest itself, right? In order to prevent, replication or anything. All right, so lysosome breaks down food. Okay, and the mitochondria, so that sausage-shaped deal, okay? Mitochondria are present in both kinds of cells because this is where cellular respiration occurs. On a quiz, that is what I would want as the function of the mitochondria. Okay, carries out cellular respiration. That is the breakdown of sugars into energy. Now, the mitochondria does have a double membrane. That double membrane acts as a bit of an insulator against the heat that is generated by cellular respiration, okay? As we wouldn't want to raise the temperature of the surrounding cytoplasm and thus possibly the rest of the cell, interfering with the rest of the workings of the cell, okay? Think about it this way. You ever had a fever? How do you feel when you have a fever? Hot. Yeah, like bagged, right? Like I can't even get up. I just don't feel like I have any energy, right? Like I'm just so tired, right? The reason a fever does that to you is because your body runs best at 37 degrees. You get any higher than that, and all of the little chemical reactions that allow you to carry out your daily business don't happen as efficiently, okay? And so it's so much harder to get anything to work. All right. If you get too hot, you run a really high fever for an extended period of time, it can be really bad for you because some of your cells will die. Okay? They simply cannot carry out the reactions that are necessary at higher temperatures. That's actually why it's a response to you being sick. Okay? Your body's natural response to an infection is to raise your body temperature in the hopes that the organism infecting you will also be made weaker by that increase in temperature. Okay, and allow you to fight it off better. Okay? Doesn't always work. Okay? It's just your body's natural reaction to it. Okay? All right, so mitochondria carries out cellular respiration. That's what you need to know about that. Okay, okay, and then the chloroplasts, right? We talked about those. The chloroplast carries out photosynthesis. That's it. And if you wrote one word for the function of the chloroplast, it should be photosynthesis. Okay. Also has a double membrane. okay, bit of an insulator. It is, after all, absorbing a great deal of light energy, okay, and carrying out a metabolic process that does generate a little bit of heat. Yeah, and you can actually see here in the description, it's talking about membranes, and then a membrane, and then a membrane, and then another membrane. Like, there's a whole bunch within the chloroplast of layers, okay, in there. Okay, so chloroplast carries out photosynthesis. All right, last of the stuff we need to talk about here. So, cell membrane. The other day I told you no diagram of the cell ever does the cell membrane justice. It always makes it look like it's made out of cellophane, okay? That it's just this layer, right? But it's far, far more complex than that, okay? The cell membrane actually looks like this. Okay? Yes, it's mostly fat, okay? That's what all these little circles are, okay? They actually have tails on them, and there's a double layer of them. So there's a layer on the outside and a layer on the inside. The tails point towards each other in the middle, okay? But embedded in the membrane, remember I said it was like Jell-O salad, okay? So there's like fruit embedded in the Jell-O. Well, that's what the cell membrane is like. There's all kinds of stuff embedded within that membrane. Okay. There are receptor proteins, there are glycoproteins, phospholipids, globular proteins, transport proteins, carrier proteins. There's all kinds of stuff that's embedded in the membrane that makes it far more complex than just this layer, okay, that surrounds the cell. Okay. Now, at a very basic level, the job of the cell membrane is to control two things. Endocytosis and exocytosis. And those are the words I would want to see on a quiz or a test. Okay? Endocytosis is movement into the cell. Exocytosis is movement out of the cell. Okay? And that's what the cell membrane controls. Okay. Now, you've probably been told before, you know, like the cell wall, the cell membrane protect the cell. And while that's, I mean, at a very basic kind of junior high level, that's an okay definition. Okay? It's not really good for high school. Certainly by keeping undesirable things out and good things in, there would be sort of a protective function to it. But the fact of the matter is things can get through. And some of them are bad. Okay? So it doesn't, you know, completely protect the cell. Its job is to control entrance and exit. That's endocytosis and exocytosis, movement in and out. All right. All right. The cell wall. Okay. The cell wall. Remember I told you I would show you a picture that made it look like it was full of holes, because that's what it is. Okay. That's a colorized electron microscope shot of the cell wall. The green part is the cell wall. The purple part is the cell membrane. And you can see that the cell wall is full of holes. It's a frame, not a wall. Okay? And it's made of cellulose fiber in a plant. Okay? In other organisms, it can be made of different things, but okay? In a plant, the cell wall is made of cellulose. And that's that dietary fiber we were talking about before. Okay? So when you eat plant material, this is the part of it you cannot digest, right? And then it goes through you and acts as dietary fiber. Okay, so if we're saying what the function of the cell wall is, it's the frame of the cell. It keeps the shape and supports the cell. Okay, keeps the shape, supports the shape, something like that. Okay. The same can be said of the water vacuole. Right? The water vacuole supports the shape of the cell, but it does it differently. The cell wall supports from the outside, and it's a frame. It uses the strength of the fibers. The water vacuole supports the shape using what's called turgor pressure. And that would be an important part of your answer. Uses turgor pressure to support the shape of the cell. Turgor pressure is the water pressure within that filled vacuole. Okay? If the water vacuole begins to empty, the plant will wilt because it is not capable of supporting itself without the combination of cell wall and water vacuole. Okay, the organelle that's only in animal cells, the centrioles. Okay, the centrioles, this is how they work. Okay, this diagram right here. So when a cell is about to divide, okay, one centriole, okay, they detach from each other and one goes to each pole of the cell. Okay, while they're moving there, they're replicating themselves because every cell has to have two of them. Okay, and so they begin replicating and they send out these fibers. Okay, so you can see them attached in this picture here, but they go out and they attach to the chromosomes that are all lined up in the middle of the cell, okay, called the metaphase plate. Just before the cell actually divides, they retract these fibers, okay, kind of like cables. So they just retract the cables in and it pulls the DNA apart, and that makes sure that each daughter cell, okay, each new cell, it has a copy of every chromosome. Actually, there's two copies of it, but it's making sure that all the DNA gets to where it's supposed to go. Right? And remember, the reason for that was that the cell could change shape during division and possibly result in the loss of some genetic material, which we obviously don't want. All right, so centrioles aid in cell division. Okay, and something else that you might see in some diagrams, okay, would be microfibers, microfilaments. Sometimes those are called the cytoskeleton, which is probably the least accurate name for them, but it's the one that's used most often. Okay, these are mostly found in animal cells, but you occasionally find them in plants as well. Okay, and they are just fibers that connect the membrane in different parts of the cell, and they kind of act like bungee cords. Right? So if the cell tries to radically change shape in one direction, it kind of prevents it from tearing. Okay? Muscle cells have way more cytoskeleton, microfibers and microfilaments than any other cell in your body because they're designed to stretch and pull and exert force and contract and relax. Okay? So they have way more of this stuff in them than any other cell in your body would. Okay? Okay, questions on any of the functions? Right? So, is that a lot to know? It is. Okay, lots of different ways you can prepare for that kind of stuff, but in the end, you do have to memorize it. Okay, I don't know if you like making like the little study cards or, you know, just rewriting the notes or making your own pictures. You got to do whatever works best for you, but know that this information is going to be required of you by Thursday.
Do you go over that peroxisome thing?
Peroxisome just contains enzymes. There's no digestion, that's the difference. But I'll never ask for like both on a quiz. Okay. If you think it's a lysosome, it's a lysosome. If you think it's a peroxisome, it's a peroxisome. Unless I specifically say there's food in that it's a lysosome. All right, I'm going to give you guys a little, let's say, 4-minute break here, and then within the classroom, obviously, and then we're going to head on to the cell transport stuff. Okay, I'll give you a quick break here.